Hi everyone, welcome. We're Team BePURE, and our project focuses on algae-based purification of landfill biogas using a CO2 removal system and a helical photobioreactor in series. So I'll be presenting for you today. My name is Anjana, and the other presenters are Praveen Putala and Melissa Meyerson. So first, we're going to begin with a brief outline of our project and presentation. We're going to start off with some background on biogas and its potential as an energy source, then go into system design based on pertinent literature review, and then we will continue with component studies looking into our systems or our methods and results, and then we will follow up with system studies based on problems and solutions, and then focus on modeling this project on an industrial scale, and then wrap up with conclusions and questions. So there's a widely recognized need for new energy sources today due to the depletion of primary sources and the fact that these sources are finite and highly polluting. Biogas is a possible alternative. Biogas is produced by the decomposition of organic material in solid waste, sewage, and farm waste. It is composed of roughly 50 to 70% methane, with the remainder being composed of carbon dioxide and trace amounts of other gases. We chose to focus on landfill biogas for our project because it is a major source and it is easily harnessable. Landfill biogas is composed of, as you can see here, majority of methane and carbon dioxide, and the remainder is nitrogen gas and hydrogen sulfide. In comparison with landfill biogas, processed natural gas is made up of a majority of methane, with the remainder being composed of carbon dioxide and other inert gases and hydrocarbons. So, as clearly evident based on the pie charts, natural gas is composed of a much higher proportion of hydrocarbons and combustible that can be burned to produce energy. So in order to make biogas a comparable energy source, we must remove enough carbon dioxide to increase the methane concentration to that of natural gas standards. Another contaminant in biogas is hydrogen sulfide, which is a toxic, noxious compound that corrodes systems, and it's also a pollutant involved in the production of acid rain. So our goal was to purify biogas to natural gas standards. In order to accomplish this, we hoped to design a system that would remove hydrogen sulfide and reduce carbon dioxide for the most part. And to differentiate from current systems, we hope to design a system that would be more sustainable with a lower environmental impact, and also aim to have this system be effective and efficient on an industrial scale. So with these goals in mind, our hypothesis was with the use of algae, we could develop a purification system that would be more cost-effective and sustainable than current purification methods. So there are several existing methods of purification of biogas. First, we can remove hydrogen sulfide through a few chemical separation methods, such as oxidation with metal sulfates, adsorption with metal oxides, or adsorption into aqueous solution of amines. Additionally, we can remove carbon dioxide through chemical and physical separation methods, such as adsorption into aqueous solution of amines, adsorption using zeolites, water and pressure methods, as well as membrane and filters. So all these methods, while proven to be effective, do have some disadvantages. They can produce waste, whether it be chemical waste from reaction with gas or carbon dioxide produced in the regeneration of material. In addition, materials such as zeolites are expensive and there are high upkeep costs. So another method, aside from chemical methods to purify biogas, are algae-based. Algae use the process of photosynthesis, as shown here, in this equation, where carbon dioxide and water, with the use of sunlight, are converted to sugar products and oxygen, which are then used in turn to propel algal growth. So we consulted literature and inferred that there were 3 algal species that would be ideal for our system, and these are Phaeodactylum tricornum, or PT, Botryococcus brownie, or BB, and Synechocystis PCC 6803, also known as SC. So these algal species use photosynthesis. For example, PT uses the C4 mechanism to metabolize bicarbonate. In this process, bicarbonate is readily taken up as soluble substrate. Our other 2 species, BB and SC, use the C3 mechanism to metabolize carbon dioxide. And here, the carbon dioxide is delivered directly to rubisco, where it is converted to oxygen. So algae as a CO2-removing medium is viable in comparison to chemical methods in that it is self-sustaining, it requires minimal nutrients, and it provides another source of energy through algal waste and algal oil as extracts from cultures. And now I'm going to hand it off to Praveen, who's going to talk about our system.
Hello everyone. So I'm going to be walking you through the overall design of our system. Our goal while designing our system was to remove the hydrogen sulfide and the carbon dioxide from the biogas and use a photobioreactor and a recycling center to recycle as many components of our carbon dioxide scrubber as possible. Our system starts with an anaerobic digester, which is used to produce the biogas that will be used for all future testing. From there, the biogas is sent to a hydrogen sulfide scrubber, which is used to remove the hydrogen sulfide from the biogas. And from then, it goes to a carbon dioxide scrubber, which consists of a water column that is used to take the carbon dioxide from the biogas and put it into a soluble solution. The resulting biogas has been purified, is then collected from our column, and the carbon dioxide laden solution is then sent to the photobioreactor, where algae are used to metabolize the carbon dioxide. And from then, a recycling center is used to remove the algae from the solution, and the resulting solution can then be put back into our carbon dioxide scrubber to make it more efficient overall. A point to note about the design of our system, we separated the removal of the carbon dioxide from the photobioreactor due to the production of oxygen because of the photosynthesis taking place. The presence of oxygen with a high concentration of methane is highly explosive. So just to avoid that issue, we separated those 2 components and we were able to circumvent the issue entirely. So the first part of our system, as mentioned before, is the anaerobic digester. We decided to construct our own anaerobic digester because it wasn't feasible for us to obtain biogas directly from the landfills. So our goal while designing our anaerobic digester was then to be able to design a system that would provide us with a steady supply of biogas, close to the composition of the biogas found in landfills. In order to maximize the efficiency of our system, we had to determine the best temperature and feed source. In order to do that, we tested 2 different temperatures. One was 22 degrees Celsius, which was approximately room temperature, and the other, 32 degrees Celsius, is closer to the temperature found inside of a cow, which is where the microorganisms in our anaerobic digester producing the biogas are usually found. We also decided to use different types of feed sources. These include soy flour and vegetable flour, and it was important to get the right feed source because they have their unique compositions of carbohydrates, sulfur products, and proteins, which would eventually determine the composition of the biogas that we would be producing. In order to determine the composition of the biogas, we use hydrogen sulfide and carbon dioxide meters, and in order to determine the rate of biogas production from our system, we measured changes in pressure. Because our anaerobic digester was effectively a closed system, any gas that was produced would result in changes in pressure. So by monitoring that, we could determine the rate of compression the biogas would produce. So shown here are the results from our first trials of the effective temperature on the biogas production. The trial in blue corresponds to the trials performed at 22 degrees Celsius, and the red line corresponds to the trials done at 32 degrees Celsius. So we can see that the rate while biogas was being produced at a relatively constant rate at both temperatures, the production rate was much higher at 32 degrees Celsius. So with these changes in mind, we made final adjustments to our anaerobic digester. We added an electric heating blanket to the anaerobic digester to keep the temperature at 32 degrees Celsius. We removed a stirring mechanism that we had in place because it was proving to be detrimental to the production of biogas. And we also chose soy flour as our primary feed source. By choosing soy flour, we were able to obtain biogas with a composition of approximately 70% methane, 30% carbon dioxide, and about 3,500 parts per million of hydrogen sulfide, which was close to the composition of the biogas found in landfills. Using these parameters, we also found that we were producing about 4 liters of biogas a day. From here, the next part of our system is the hydrogen sulfide scrubber. For this portion of our system, we decided to look at the previously established methods. This was both due to time constraints and also because hydrogen sulfide, as Sanjay mentioned, is extremely toxic and corrosive, and especially to metallic components of our system. So we wanted to make sure that we got rid of as much of it as possible before sending the biogas to the next portion of our system. We looked into 2 well-established methods. One used a copper sulfate solution so that when the biogas was bubbled through, it would produce copper sulfide, which would then be precipitated out of solution. Another method involved having a column packed with rusted steel wool so that when the biogas was sent through, it would react with the iron oxide to form iron sulfide. And we looked into both these and measured their efficiencies by measuring the hydrogen sulfide concentration before and after the biogas was sent through the columns. Ultimately, we decided to use the rusted steel wool method. This was for multiple reasons. The copper sulfate solution was proven to be expensive not only to get the copper sulfate, but also to dispose of the copper sulfide properly. We also ran into an issue with the pressure, where the pressure required to bubble the biogas through the copper sulfate solution was actually more than the pressure provided by our anaerobic digester. It should be noted that the lack of enough pressure is not due to the microorganisms themselves, but probably due to faults in the design of our anaerobic digester. By observing our steel wool column, we were able to see physically that the proper reactions were taking place. And when we measured the hydrogen sulfide concentration after the biogas had gone through, we saw that it had less than 1 part per million of hydrogen sulfide. So based on these, and although both ways work well, we chose to use the steel wool column just because it circumvented the issues that we ran into. The next part of our system is the carbon dioxide scrubber, and for this, we built a water column. As we pictured here, we can see the water goes in through the water import and exits through the water outport. Same as similar to the gas, which is sent into the gas import and then out through the gas output. To improve the efficiency of the carbon dioxide scrubber, we also added ration tanks, which serve to break up the biogas into smaller bubbles and increase their surface area, which we hope would increase the efficiency overall. We decided to test the effect of the pH of the water, the aqueous solution that was inside the water column, and the effect that it had on the efficiency of the removal of carbon dioxide from the biogas, which can be seen here. We noted a pretty sort of cool relationship of the higher the pH, basically, the more carbon dioxide we were able to remove. So that at a pH of 12, we were able to remove about 85% of the carbon dioxide. However, we ran into an issue again with the pressure, where the pressure required to bubble the biogas through the water column was higher than what the anaerobic digester was able to provide us. In order to get around this issue, we designed a spray column so that instead of bubbling the biogas into the water column, we could instead spray the aqueous solution down over the biogas. Also, following the previously observed trend between pH and carbon dioxide removal efficiency, we increased the pH of the aqueous solution to 13. After we made these adjustments, we noted that we had approximately 11% of the carbon dioxide remaining in the biogas after it had gone through our column. It should also be noted that for the carbon dioxide testing, or the scrubber testing, we used compressed carbon dioxide and nitrogen instead of the biogas produced by our anaerobic digester, mainly due to time constraints, because we did not want to be waiting for our anaerobic digester to be up and running before we tested the efficiency of the scrubber. This had its advantages, which provided us with a steady concentration and composition of our gas as it went through the column. However, the flow that was provided by the compressed gases was much higher than what our anaerobic digester provided us with, so we ran into that issue eventually as well. So our initial testing of the new spray column yielded some unexpected results. You can see from the blue line here, there's unexpected drops and rises in the concentration of the carbon dioxide as it goes through our column. So we did some troubleshooting and found out that the position of the sparger that bubbled into the biogas was actually below that of the water output. So this was allowing some of the biogas to leak through the water output, and also it was interfering with the ability of the carbon dioxide meter to measure the concentration properly. So we simply raised the position of the sparger to be above that of the water output, and noted a much more steady decline in carbon dioxide concentration as it went through our pump. I'm going to be passing it off to Melissa. We'll be talking about the algal portion of our system.
Hello everyone. For the algal portion of our system, we looked at growth conditions for two of our algal species, the BB and PT. We examined different stirring methods, including rocking, vegetation with the incubator, and centrifugal and diaphragm pumps. We also looked at the effect of the addition of sodium bicarbonate and sodium hydroxide on the growth of the algae. And these are both important because the addition of sodium bicarbonate to the algae provides it with an extra carbon source, which allows it to grow better. But sodium hydroxide was an important test because our CO2 laden solution contains a high concentration of sodium hydroxide, and we needed to ensure that the sodium hydroxide would not kill the algae or harm it in any way. We made measurements using optical density at 600 nanometers, which measures the absorbance of the algae, so we can determine the concentration. We found that in terms of the sodium bicarbonate trials, the higher the concentration, which is the red line, 100% of maximum concentration coming from our scrubber, yielded better growth. The PT showed a similar trend after about 15 days. However, after that, the growth dropped drastically. We think this is possibly because of contamination of our culture. It was at this point we discovered that the initial culture we had obtained had been contaminated. We obtained a new culture, however, we did not have time to repeat this step. For our sodium hydroxide trials, we found that both algae maintained a relatively constant pH of 7.5 or 9.5 for BB and PT. This indicates that there is some buffering effect, and the algae are keeping the pH of their medium constant, even though high pH solution is being added. We also found that the centrifugal pump did not work well because it was too strong and killed the algae, although our other stirring methods worked fine. In order to integrate the algae into the rest of our system, we created a helical photobioreactor. It's composed of a helical coil here, connected to a two-part reservoir. The reservoir is in two parts because one part is used to add the CO2 laden solution to the reservoir. The other part is used to vent the oxygen that is produced by the algae, as well as recycle the excess algae to the recycling system. We use the coil design because the high density of algae, which is seen in this picture, prevents light from penetrating. There's a light in the top corner. However, the density of the algae prevents it from going into the entire culture. The helical design avoids this problem because it spreads out the algae, making the density low enough that the light can penetrate all of it. We made qualitative observations of the BB in this system and found that the algae stuck to the walls, which are the green in the picture. We also found that the tubing crimped and would not stand up straight, and our pump failed. In order to fix these problems, we switched from the BB to the PT, which should not form the films that we saw here. We also decreased the diameter of the tubing to increase the turbulence and prevent crimping problems, and we found a new, stronger pump. So now I'm going to talk about our modified system.
So here's a picture of our modified system. First, we have a heated digester without stirring to improve the production rate. Next, we have an H2S scrubber steel wool-filled column, as well as a CO2 scrubber spray column with pH 13 solution, both of which exist to circumvent the pressure issue previously mentioned, where the pressure required by the water column was greater than the maximum pressure produced by the digester. And to summarize the flows within the system, the majority of the water flows into the CO2 reservoir, with the remainder going into the algal reservoir. From here, the water flows into the photobioreactor system, which is shown here as this is the photobioreactor. The reservoirs are not shown in this diagram. And then from there, the excess water goes into the recycling column, and a filtration process occurs where it's filtered of algae and then finally comes to the CO2 reservoir. And so we calculated the system parameters, which include the flow rates and column size, through the growth rates of algae and the carbon dioxide content in the system. And we also consulted literature and decided to use a lighting value of 1440 lux for our experiments. So in order to test this modified system, we inoculated the culture, adjusted the pH to about 13, measured the biogas composition coming out of the digester and the CO2 scrubber, and we measured algal growth through optical density readings and dry weight measurements. In testing the system, we ran into a few problems. We noticed that after just 2 days, the salt from the instant ocean that was used to make our media formed a white precipitate after reacting with a sodium hydroxide solution. And this clouded the media and reduced illumination. Moreover, this affected our growth measurements and also clogged the valves in our system. Additionally, we observed that we did not add enough algae initially to our system, so we observed inadequate algal growth, and the pump also stopped functioning normally during our experiments. So in order to address such issues, we chose to use a freshwater algae called Synechocystis that did not form filaments. We decided to start with a higher density of algae to improve growth and have a buffering effect, and also you went with a more durable pump. So with these adjustments and the new algae, we took measurements once a day, and we noticed that the Synechocystis algae stuck to the walls. The pump also failed as it tore a hole in the tubing, which caused the sodium hydroxide solution to leak. And so due to time constraints, we were unable to fix this pump issue, and we also could not make fresh media. So unfortunately, our algae died due to an increase in pH to about 10.8. So based on these results, we could conclude that our adjusted system did not work as we'd expected, and the new algae did not have the same buffering effect as the other species. Now I'm going to hand it off to Melissa to talk about scale-up.
Alright, after conducting our system tests, we did a scale-up and cost analysis to determine if our system would be effective on an industrial scale. These are based on the average biogas production of a landfill in Maryland, which is about 1 cubic meter per second, and has a composition of roughly 70% methane and 30% CO2. The volume shown in the middle is the volume of liquid that would need to be in our system to completely remove the CO2 that's being produced. This is an unfeasible volume for a system to be too large, so we calculated based on a system with these dimensions and found that we would need this either 23, 39, or 82 systems of that size to completely remove the CO2. We then calculated the revenue that would be produced from our methane and algal oil, and found that it's between 4.93 and $11.6 million per year, depending on which algal species is used in the system. We also calculated the cost of upkeep for our system, which includes the cost of sodium hydroxide and the electricity to run the pumps and the lights. The total cost for our system would be between $14.5 and $64.5 million per year, again depending on which algae is used. After that, we combined the revenue and cost to determine what our total profit would be, and found that the cost outweighed the profit and outweighed our revenue, and therefore we would be either 2.9 to $57.4 million. We'd be losing that much per year with our system. There are some ways we could help reduce this cost. We could consume less sodium hydroxide, which would lower the cost. We could also use more efficient lighting, and we could produce more algal oil, or we could integrate or produce natural gas to power our own system. From this scale and our system testing, we can conclude that although this system is not a more cost-effective and efficient method to purify biogas, it did successfully produce biogas that was close to natural gas standards. There are several future things we could do, including system modifications, which would be using different algal species. We could also insert backup pumps into our system so the pump failures we experience would not prevent the system from functioning. We could also investigate alternative methods to dissolve CO2, such as increased pressure and decreased temperature. We can do other experiments, which include biological removal of hydrogen sulfide, as well as metabolic engineering of the algae to determine if growth rate can be increased or it can reduce from olive oil. And finally, we'd like to acknowledge everyone who assisted us with our project. Thank you so much for your help.
As part of the time, I'm going to ask discussants have 10 minutes to address the BePURE team. And then after the discussants, we're going to ask for anyone in the audience if you have any questions that you would like to ask the team.
You guys consider using sunlight in your CLO process? So you can take to create meaningful light outside.
So the question was if we consider using sunlight instead of artificial lighting in our scale system. We have actually kept with our current system of keeping the artificial lighting in the middle of the photobioreactors. And the main reason is because once it's outside, there's completely different observations and findings that you're going to get in terms of results. And also to keep it consistent with our data and the different things we've done in the experiment, we decided to keep going with artificial lighting. So if we're using a sunlight instead of artificial lighting, definitely the cost will be a little less. But in terms of the amount of cost that we've seen for the lightings, I believe the majority came from other places, so we don't believe it will be a huge factor in the scale up.
One more general question. So, if you have biogas from the landfill, what can you do with that biogas directly? And the cost of that and the profits associated with that versus doing the H2S scrubbing, the CO2 scrubbing, the bioreactor, kind of each of those processes and the cost benefit of each process. You're showing this as a total package, but can the cost benefit of this analysis work if certain components are
So the question is, what are the benefits of doing these purification steps rather than just running the current things that we're doing with just the biogas alone? From the literature, from what I've read, there's a lot of instances when you just flare the biogas directly. The problem with that is the energy content is not the highest it can be in terms of composition. As we said before, biogas is roughly 50% methane, maybe sometimes higher, and roughly 30 to 50% CO2. Carbon dioxide doesn't have much energy content, so when you combust it directly, the biogas, you're only going to get 50% that combusting giving energy. And the reason why we have to go through with the H2S scrubber is because it's highly corrosive. The amount of maintenance costs that will be implied because you don't purify the H2S out of the system, it'll actually pose more cost in terms of maintenance later on. CO2, I think I've addressed it, and that's to get rid of the excess gas that does not have any energy content into it. And the reason for algae is the reason that we have the part that we did not get to. It's the algal oil. It adds a lot more profit to it. So especially the first algae that we use, the BB algae, we can produce up to 70% algal oil, and using metabolic engineering or different factors, it can definitely add to the revenue rather than just having to get.
My concern is about the coordination of this microbiology. This is a high pH because we have to use a pH swap. High pH opens the water for growing algae. They tend to aggregate, they tend to stick to the wall. It's very common issue. Maybe another modification would be to use compressors on the CO2 reactor.
one of the major issues with putting the biogas directly into the algae system is that the CO2 is moved through photosynthesis, and photosynthesis produces oxygen. So if you're collecting the gas from the algae system directly, you're going to end up with considerable oxygen in the mixture as well. And that is a major mixture of applicator remove the oxygen before you can use it
Initially, we did discover that the pH caused many safety problems in every system we tested by them. That's why in future directions, we think it would be a good idea to look at systems that don't use sodium hydroxide. CO2 can also be dissolved if we increase the pressure of the system by pushing out the first one. They're all different.
Not a possibility, maybe just keep it in all those species as well.
Yeah, we do that.
So this is the CO2 remaining. So before with the race merger, you had you went from 45% down to 30%. So you're getting about 10% removal of the CO2 in the system. So I'm wondering, and that contributed a lot to your cost with all that stuff. So I'm wondering if it's worth, again, kind of going back to my first question, is the algae, again, you're raising the pH so high by doing the CO2 removal process, and you're only actually getting about 2% of the CO2 out. So is this really cost effective for that? It's here.
Our analysis shows that this method, as it is not cost effective. For many reasons because of the energy price. So again, several things need to be changed in the system in order to make it possibly cost effective. And that includes dropping the pH, that eliminates the cost of adding sodium hydroxide. It eliminates a lot of problems in the system. And if we can use a different method of dissolving the CO2 in the aqueous media, and that would greatly reduce the cost of efficiency of the system.
One of your decision criteria is 1.2 psi, the gas pressure.
Directly collected from a landfill, the pressure would necessarily be extremely high, but that gas can be easily compressed. And so if we were able to compress the gas at a time, we would be able to change the design to potentially a much higher CO2. For example, higher absorption column and blow the gas through a large water column was more efficient than the spray column. Yes, that was a decision out of necessity, not out of ideal circumstances.
Second question, how do you get the algae out of the system?
So the question was, how do we get the algae out of the sand filter? And so our sand filter, basically what it does is that it traps the algae at the top of the layer, allowing more algae to build up at the top, whereas allowing the clean water to flow through the bottom of the sand filter. And so to remove the algae, although we didn't have it in our project, we would recommend using a clear PVC piping to see how much of the algae is built up. And usually after a certain period of time, say a few weeks to a month, the algae would build up to a point where you could just scoop off the top of the sand layer and then replace it with another layer of sand to remove the algae. Well, our current plan was to, as with our project, was to remove remove it, since most of the algae will be dead by the time we collect it. Our other option, which we didn't get to, was using the algae for algal oils, which could also be a potentially profitable asset.
Does that require energy?
Yes, it does require energy, and yeah, we'll definitely look into that. And that was something that we were planning to look into, but we didn't get enough time to.
So what challenge do you see on is it the order of the output of the small box that's so insufficient micromanagement? So the spirit of evolving, the experience of challenges in getting a comparable reactor that is still needs to have the right process for accomplishment.
Yes, there are many issues with trying to design a photobioreactor that would be able to handle the amount of biogas produced by landfill and still be reasonable. We kept a small tubing size of diameter 0.1 meters in order to maintain surface area so that light actually can penetrate through the entire tubing. And then we limited the height of any coil to about 100 meters to avoid issues. And then we replicated these coils with our farms. We also kept the diameter down to, I believe it was 5 meters, 10 meters per coil. 25-meter radius coils so that the lighting incentive didn't have to have enough intensity to increase that. In terms of the cost analysis itself, the dimensions of the helices are relevant for total production planning costs and costs. Okay, construction costs are initial costs. So the initial costs are not covered in our cost analysis because it's extremely hard to determine it's very difficult to determine accurate costing for a sort of newer system. So we pretty much just looked at the upkeep cost to determine if eventually, regardless of how much construction cost we have, we would pay.
So the question was whether our photobioreactor design is comparable to any existing designs that are out there. The previous steps with the H2O scrubber and the CO2 scrubber, there are previous designs that we could use and look into. But because our main focus was the photobioreactor, we didn't really look into those type of costs. In terms of photobioreactor, it's still fairly new. From what we see in the literature, review has grown exponentially in the last 5 years in terms of these. And they mostly use an airlift photobioreactor system. It's the helical design. And that is where you follow the biogas directly. But there are challenges with that as well. In terms of the size that we're looking at, this size is of roughly 20,000 liters, if I'm not mistaken, is much, much bigger than the design that the research level is looking at. So in terms of that, we're not able to calculate the capital costs. Does that answer your question?
Yeah, this is a very exciting project. You integrate different technology from chemistry, biology, physics, physical process. And based on your calculation, at the end, if you start a business today, a year later, you're going to be losing like $20 million, $50 million. So, you always a very high student. Tell me what you're going to do to improve.
There's several things that you could do. I think some of them were addressed in the future direction, and I'm going to ask the team to kind of think of more ways as I kind of go on with my much algal oil that you can as possible. The other thing is to reduce the amount of NaOH that you use. The NaOH cost is significant, quite frankly, so if you can use pressurized water system with a CO2 absorption column, then that cost would be mitigated to a degree. And the other easy thing you can do is find better algae, get better growth rates. That way, the resident time can decrease, hence the volume can decrease as well. That reduces the capital cost and the upkeep cost as well. And the lighting, to get that as efficient as possible to reduce the amount of heating that can happen, and also to put it outside where you can use the sunlight rather than artificial light to that adds cost to the system. There are various different things you can do.
We ran many trials for every experiment that we ran. This is only displaying one trial because the data was shifted in the trials in terms of time. However, these trends were observed several times. So
those products are always took up significant amounts of CO2 a day. Put it back.
yes, that's how we discovered the issue. Was we found it unusual that it would that the CO2 concentration in the effluent gas from the CO2 column would drop so substantially compared to our previous studies, and then go back up. So we recorded basically the time that we did everything, and the first major drop is when the water reached the bottom of the column, and when the water started coming out of the water outport, which is when we opened it. And that's where we believe that we observed gas then coming out of that tube. And because gas was coming out of that tube, then instead of going into the rest of the CO2 column, then the CO2 meter, which had a pump built into it, generated negative pressure. And so we believe that that might have been enough to overcome some point in our column and pull in atmospheric gas, which the composition of CO2 is close to that of atmospheric gas. In most of our studies, we use a CO2 meter, which I'm not entirely sure.
I think Vicky's going to ask us to wrap up a little bit, so we can kind of address that during our discussion feedback time.